Hello everyone and welcome to the first of the Superbio Masterclass series. My name is Catriona Nguyen Robertson and I am a Science Communications Officer for the Convergence Science Network. Before I begin, I would like to acknowledge the traditional owners of the lands on which I'm streaming from, but I recognize that you might all be coming from different lands across the state, which is absolutely awesome. But I myself am on the lands of the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. And I think it's important that we remember that they are the first scientists of this land in their endeavor to understand the land, the sky, and the sea. And science, that's why we're here today, because we all love science, especially biology. And I might be biased, but I'd say that biology is one of the most exciting fields of science, and it's critical to our understanding of life. Biologists play a major role in growing our knowledge of biological processes and in translating that knowledge into therapies and treatments. The Superbio initiative was established to share the wide range of careers that biology can offer for all you VCE biology students, or maybe those of you who want to pursue VCE biology. And I'll introduce tonight's speaker, Jess, shortly, but I just want to say that biology can really take you in many different directions. Both Jess and I studied science and we undertook PhDs in biomedical sciences, and now she's a researcher researching fundamental questions in biology, and I've kind of moved into the education space. So there's really a wide range of careers that you can get. So in this particular series, the Superbio series, you'll meet biologists who are working across academia, research, business, and each of them are really taking their passion for biology and making an impact. The Superbio initiative was developed by Monash Tech School, one of the 10 tech schools in Victoria that are really championing future skills with future research and industry capabilities. It's delivered in partnership with the Convergence Science Network, which is why I'm here. And the Convergence Science Network engages the public with advances and innovations in the biomedical sciences space. We're also really grateful to have the support of the Victorian Department of Education to deliver this initiative. Now, on to the main event. It is my absolute pleasure to introduce Dr. Jessica Greaney, or Jess, who's a research scientist at Monash University in the Australian Regenerative Medicine Institute. Now, hi Jess. I don't want to steal too much of your thunder, but briefly, Jess is a first-generation tertiary student, so no one in her family had gone to university before, but Jess was really fascinated by biology at school in rural Queensland, and then moved to Brisbane to study biomedical sciences. And now we're very fortunate to have Jess here in Melbourne as a researcher studying teeny tiny embryos. And we're going to hear about your research in a bit. But I'm just going to throw in there that Jess takes award-winning pictures with microscopes. So Jess, I want to start all the way back at the beginning. What sparked your interest in science? I hear that you had a toy microscope.
I did, yes. Thank you for the intro, Catriona. So I was kind of interested in science from a really young age. I can't sort of pinpoint exactly when that happened, but I remember someone who had a particularly strong influence over me when I was quite young was actually my grandfather. He was very interested in gardening, and I sort of used to wander around with him in his garden looking at all of the insects. He had a hive of native bees that I remember I used to spend hours just watching them and seeing what they were doing. And, you know, he would teach me all of the Latin names of all of the species of the different plants in his garden. And, you know, we would spend hours kind of wandering around looking at things and seeing how they'd changed, if there had been rain or if it had been quite dry recently. So I think spending time with my grandfather definitely kind of piqued my curiosity, I guess, about life and the world around me. And then my parents were very supportive of that, even though, as you mentioned in my introduction, they're not scientists themselves. But they were very encouraging of my curiosity and very supportive of kind of my interest in all things life and the world. So I had a toy microscope when I was quite young, and I would walk around the garden picking up various different things like insects or I would take scrapings of the inside of my cheek or steal hairs out of my sister's heads to try and look at them under the microscope.
I bet
So
she loved that.
yeah, she did. They're very patient with me.
So that's interesting that you've kind of moved from studying insects and plants under like a toy microscope to now looking at, well, you're still in biology, but I guess now you look at humans, and maybe you started off with your sister's hair. Um, so you had your grandfather who kind of nurtured that real love of science and that want to ask questions. Did many people around you pursue science? I know that you were sort of in more of a rural town.
Yeah, so the town that I grew up in was called Gympie, and it's in Queensland, in the southeast corner of Queensland, kind of a few hours north of Brisbane. And it's quite a small town, and there really was no scientific industry there whatsoever. It was quite unusual, I think, to be interested in science. And definitely to pursue science as a career. Both of my parents, neither of my parents finished high school, as was quite common at the time, especially in small towns. They left high school in year 10, and my dad went on to become a printer, and they owned a printing business. So they would print things like business cards and wedding invitations, newsletters, that kind of thing. So they never actually graduated, and never thought about going to uni because it just wasn't really a common thing back then. But I mean, both of them were very intelligent people. They were always really interested in reading and, watching documentaries. So they were very interested in learning. I think it just wasn't really an expected thing that they would go to university. So it was quite unusual for me to want to pursue science and to go to university. I was kind of the first one in my family to go to uni and actually be interested in science. But despite them not really understanding that world, they were always super supportive of it.
And if it was unusual for people around you to kind of go to university, I mean, presumably your teachers did though. So did teachers at school help you along the way and influence your decision to study science at university?
Yeah, definitely. I went to quite a small, very Catholic school, so there wasn't a lot of sort of dedicated science classes, but I did still have a few teachers who kind of made an impact on me in that they were very encouraging of my love of learning. So I had a few teachers in primary school that really encouraged me to be good at problem solving in terms of like mathematics problems, and really into reading and spelling and that sort of thing. Then when I moved into high school, and we started doing more dedicated science classes, there were a number of different teachers there that were quite influential. And I remember this one teacher in particular who was quite a character. He was this kind of big, larger than life American, very, very loud voice, kind of really lovely guy. And he brought in a set of cow's lungs one time into our science class, and he attached a vacuum to the lungs and was kind of filling them up with air. And it was just one of the craziest things that happened at school. And it was so interesting and so out there that it really drew my attention.
Yeah, I can imagine. And that's like in addition to, I don't know if you did a lot of dissections in school. I know you'd have the heart, you'd have the bullseye and all that kind of thing. So to see a lung like probably inflating, deflating, that would have been really cool.
Yeah. And a lot of cane toads, because there are a lot of cane toads in Queensland. So we were doing cane toad dissections as well.
Is that sort of like cane toad population control? Put them in schools?
Yes. I think inadvertently it was probably a good thing environmentally.
But aren't they poisonous as well? Like
they are, but they're not really jumping around while you're dissecting them. So I think it's safe at that point.
The things we do for science. Speaking of the things that we do for science, I believe that you had a work experience gig in pathology. So what did you get to do as part of that? Was that part of like a year 10 work experience type of thing?
It was, yeah. So I wanted to do work experience in a lab. I knew that I was interested in science, and I kind of spoke to my guidance counselor at school and told them I'm interested in science and, what can you do? But living in Gympie, there wasn't kind of any research institutes around for me or universities around for me to get that experience in a research lab. So the kind of closest next best thing, I guess, was to be in a pathology lab. So that was based in the Gympie Hospital, and it was in the basement of the Gympie Hospital, and we would have all of the samples come down a pipeline in the building. So they would pop down into the box down in the basement, and there was all sorts of lovely things, different samples that we would collect. And I spent quite a few hours making blood smears. So we would take blood samples and smear them across a slide, and then we would look at them under the microscope, and you could see, if you add different dyes, you could see all of the different cell types. And I was kind of responsible for making the slides and then doing some of the counting of different cell types. We also made up some like antibiotic plates so that we could put patient samples on the antibiotic plates and see what antibiotics could be used to treat their infections. And I really enjoyed the technical aspects of being in the lab, so making the plates and making the slides and looking down the microscope. But it was kind of my first experience of a clinical setting, which I think made me realize that maybe that wasn't for me to work in a clinical setting, because I think it was quite confronting at times seeing the patients, and I much more enjoyed the technical aspects of the lab work. And it was
Even though
my
you
first.
were hidden in the basement?
Yeah, I preferred being in the basement away from from everyone else. Yeah.
Yeah, it certainly sounds like an upgrade from the toy microscopes. So that was obviously during high school and at kind of a pivotal time where you're deciding what sort of subjects do you like to do, and maybe you realize then that you didn't want to, maybe be in a clinic, but you still wanted to pursue science, right? So what subjects did you take in high school? Because a lot of students here are, currently making these sorts of decisions. And how did you make the decisions of what subjects to take?
Yeah, so I knew that I wanted to do science. I knew that I really, really liked maths as well. For a while there, I kind of considered maybe doing maths at uni, but I had no idea what kind of career that would eventuate into. Science was a bit more simple. It would be to be a scientist. So I kind of spoke to, well, actually, first I took a test at school that was, I think it was in about year 9 or 10, when we were kind of looking at picking our senior school subjects. And I took a test at school where they kind of asked a lot of questions about, what are you interested in? What kind of career do you want? And I got my results that I should be a scientist, which I already knew. So there was the option of biotechnology or biomedical science on as my test results. And so then I was advised by my school's career counsellor to do science and maths. So at my school, I'm not sure if this is the case in Victoria, but at the time at my school, we had the option of doing biology, chemistry, and physics, but you could only pick 2 of the 3. So just, I think, in terms of timetabling clashes. So I was only allowed to pick 2 science subjects. So I picked biology and chemistry, mainly because physics was a lot less interesting to me. I found biology and chemistry quite cool, and I really enjoyed the labs and, mixing things together or cutting things up in the labs. So I thought biology and chemistry were a good fit. I also, in about year 10, I think, looked into the possible degrees that I could do at uni, because I knew that I wanted to go to uni. And I looked at biomedical science or a Bachelor of Science, and I saw that, often you have to have a certain number of science classes. So I think you had to do a minimum of 2 science classes, which I was already doing. and you generally had to do Maths B, and I chose to do Maths C as well. So those are kind of the advanced maths classes. And I also spoke to my parents to kind of see what I was allowed to do because I wanted to do drama because all of my friends were in drama class. But my parents asked me why, and I didn't have an answer because it was just because my friends were in drama. So I chose biology, chemistry, maths B and C, and some of the advanced English courses.
I guess that's sometimes important to consider that, our parents can have an influence on what subjects we do. Ultimately the choice is yours, but, definitely acknowledging that parents can have a say. I would agree with the biology and chemistry. That's not to say to all of you out there that, physics isn't worthwhile. It's just what you're interested in, right?
It just wasn't for me.
Yeah, same. And sometimes biology can be paired really well with physics. So, if you love it, go for it.
I ended up actually doing a biomedical physics course at uni, and I could have used a lot of the physics basics that I didn't have, and I kind of had to learn them on the fly at uni, which was quite difficult.
Yeah, I was actually the same, and I had to kind of do a fundamentals course at uni. So we both ended up doing physics anyway.
Yeah.
So you've mentioned your career guidance counselor a bit, and your parents and some teachers as well, and your grandfather, of course. So you've had these mentors early in your career, or rather, like while you were still in school and making quite big decisions. Do you still have mentors now, and are they the same? I wonder if students here find that they have people that they can look up to as role models, or people that they can turn to for mentorship and guidance. What is the role of a mentor as you progress, and who are your mentors?
Yeah, so I think mentors, and I'm beginning to realize how important they are. I have had multiple mentors over the course of my kind of time as a PhD student and now as a postdoc. But as you mentioned, looking back, I still had people as a high school student that I would seek advice from, or kind of just have them as a sounding board for my ideas. I guess that's kind of what my parents were, because they themselves didn't really know a lot about science as an industry or university and the process of enrolling in uni. But it was good to have them there to kind of tell them what my ideas were, and they would maybe ask me for more details. So that would make me think, oh, maybe I need to research this a bit more. I don't really know what I'm talking about. So I need more information. So now I have mentors kind of for the same purpose, I guess. I have mentors that I give updates kind of on where I'm at in my career. We talk about what my goals are for the next, 5, 10 plus years. And we try and have a discussion of what I'm doing now that can help me in the future. Because I don't want to get 5 years from now and go, oh, where are my goals? I haven't achieved my goals. I need to be working at them 5 years prior, if that makes sense. So I use my mentors to kind of give me a bit of a boost and give me a bit of, I guess, encouragement, but also clarity in my ideas and coming up with a plan.
Yeah, mentors are absolutely great. And I do want to touch on your 5-year plan later. We'll talk about the future later, but just continuing on with the past, because that's where most of our audience are right now. How did you go about choosing a degree and the university that you wanted to attend when you finished up high school?
Yeah, so I kind of, I remember I had a few options in mind with what I wanted to do for my degree. So as I mentioned, I was kind of interested maybe in biotechnology. I ended up doing biomedical science because I'd sort of spent hours and hours going through all of the possible material online on university websites where you can sort of read about the different degrees and what they might what information they might include. Biomedical science sounded appealing to me because it was research focused, whereas biotechnology was more of a kind of technical career trajectory. I was more interested in going into research. So biomedical science was the one that I chose. It was quite similar to doing a Bachelor of Science. The difference for me was that a Bachelor of Science was a bit more of a broad kind of course, whereas biomedical science was very much for people who wanted to end up in research. So that was kind of the area where I wanted to head, and then I had to choose a university. So I wanted to move to Brisbane because I wanted to get away from Gympie because it was a little bit boring as a teenager. But also, there were other closer unis like on the Sunshine Coast, which was only about an hour and a half away from Gympie. But I wanted to go particularly to UQ, the University of Queensland, because I could kind of get a gauge that they had quite a good reputation for research, and that it was quite a research-focused university. The way that I could kind of gauge that was looking at the research institutes that were on campus. There are a number of different places like the Queensland Brain Institute, the Institute for Molecular Biology. So I knew that UQ seemed to have a lot of the facilities to do research. And then I read about university rankings and how they were ranked highly in different areas, and science was one of them. So then I asked my parents if I could attend the UQ Open Day. And I think I went to that when I was in maybe year 12. But I think you can go earlier if you're interested in like year 11 and 10. And that really sealed the deal for me, was going to campus and seeing how everything was. It was such an amazing kind of experience to see, a real university campus and see how big it was and how, exciting it was with all of these different facilities, and the classrooms looked amazing and the labs looked amazing. So, so that kind of sealed the deal for me that I chose biomedical science at UQ.
And obviously, you just pointed out that the classrooms were amazing and the labs and all the facilities and things. So university life is quite different to school life, especially if you've come from a small town. So what was it like studying at university right after high school?
I found it quite different in a lot of ways. In high school, I think you have a lot of accountability to your teachers. So if you haven't done homework, for example, your teacher is telling you you need to do this homework, or asking you why didn't you do the homework. Whereas university is quite different in that it's very self-motivated. You have your timetable. So you'll generally have a timetable where you'll have lectures on at certain times, and then you might have a tutorial, which is more of so lectures are quite big generally, a couple of hundred people in the room. And there's a professor at the front who is just running through the material. You might have time to ask questions at the end, but really it's quite uncommon to ask a lot of questions during a lecture. So then you would have a tutorial where you're split into smaller groups, and you are often given exercises that are kind of like the homework that you have at high school, where you're working out of a textbook, or you're working on particular problems that you have to solve together. And then you also have, if you're doing a science course, you often have practical classes where you're in a lab. So you get a lab coat and you put gloves on and you work on whatever experiment it might be. It might be, dissecting a toad, or it might be, doing some genetics screenings or something. So if you choose not to go to your lectures, there's kind of no one there checking on you. And that can be a good thing and a bad thing. A good thing in that, it teaches you independence and it teaches you to self-motivate yourself. And it can be a bad thing if you skip too many lectures and then you have to kind of catch up later on during exams. And I think generally in lectures, the lecturers don't know you by name like your school students or your school teachers would. So you're kind of a bit more of a number in a room to them. But it's quite easy to make a lot of friends at uni, I think, because you are, spending that time in the smaller groups in your tutorials and in your practical classes, and you're working on group assignments together. So it's really easy to meet new people. That was something that I was kind of worried about when I was, moving to a different city and leaving all of my school friends behind was, how am I going to make friends again? But it was super easy because you have so much to talk about because you're all kind of studying the same thing and you have the same assignments and the same exams coming up. So, it's quite easy. You have a lot of opportunity to meet people and to make friends. And I think, when I first looked into going to uni, I was thinking, gosh, 4 years is a really long time. I don't know how I'm going to get through 4 years of uni. Like that's a really, really long time. But I ended up doing 8 years at university, and honestly, it went by so quickly, especially the first 4 years in my undergraduate. It went by, yeah, and I found it quite enjoyable. Obviously stressful at times when you're in exams, but I found it overall very enjoyable.
Yep, exams are stressful. That's
Yeah,
I guess that's part of
no way
it. But
around that.
I'm guessing that I'm just going to make an assumption, but I'm guessing that a lot of people here on this call are quite motivated, given that you're here on it, a weeknight.
Yeah.
So yeah, hopefully you find that university isn't too much of a jump. So you've mentioned that there are lots of research institutes attached to the University of Queensland, and as a second year student, I believe that you did a research program or project at the Queensland Brain Institute. So what did you do as part of that, and did that sort of solidify your resolve or your want to do research?
Yeah, so that was actually part of kind of an extracurricular program that I was involved in. So it was called the Advanced Study Program in Science. And that was something at UQ that was designed for students who got a good OP score, which is, I think, the same as like an ATAR score. So if you got a good score at the end of high school, you were eligible to apply to an advanced study program in science. Basically, it was a program outside of my actual courses at uni where we would kind of meet in our spare time and we might have a scientist come and talk to us and tell us, about their work or kind of what how they got where they are in their career. But we also had an opportunity to do kind of a very small research project. So I believe it went for about 6 months. And I was based at the Queensland Brain Institute, as you mentioned. So when I first went in, it was my very first experience in a real research lab ever. And on my first day, I watched a rat brain surgery, which was very unusual. Very cool, but very, very unusual. So they were, I think, implanting electrodes into the brain of a rat to sort of study the electrical impulses in their brain. So that was really interesting. My project was actually looking at the vitamin D levels in decades old blood spots. So when a newborn is born, they would take a heel prick. So they would prick the heel of the newborn and spot a little tiny spot of blood on a piece of paper. And what I was doing was taking these blood spots from, 20 plus years ago and cutting little sections of them out. And we would then run them through testing to see if we could measure their levels of vitamin D. So the project was looking at when a mother is pregnant, if she's getting enough vitamin D or if she's not getting enough vitamin D, is there a correlation between low vitamin D intake and developing schizophrenia later in life? So the lab that I was in was particularly interested in schizophrenia. So we were looking at all of these blood spots and then correlating that to whether these people eventually ended up developing schizophrenia later in life. But it was very interesting. But I actually didn't enjoy it as much as I thought that I might. And I think it had a lot to do with the project itself. It was kind of boring, and I don't really like blood. I also didn't really have kind of a go-to person in the lab, so I wasn't sure, what I should be doing, and I kind of sat at my computer a lot. I wasn't really sure, where I'm supposed to go and what I'm supposed to do. Being in a research lab was quite a different experience for me. Um, but yeah, I sort of thought that was interested in neuroscience initially, but maybe I wasn't as interested as I thought I was. But
And
I
that's
think,
okay. It's okay to
yeah,
change your mind.
I think it's a good thing. If you don't like one aspect of biology, there are so many different areas of biology and so many different smaller fields that you might find something you like outside of that. So it wasn't too disheartening that I didn't really like neuroscience, but I knew that I really liked science in general and biology. So that kind of pushed me to try and think of something else that I could do that wasn't neuroscience related.
Yeah, so I mean, it's okay to jump disciplines, and which you did to do honors. Now, I don't want to get too much into what you did in honors in terms of the science, because I really want to hear about what science you're doing now. But just so that people understand your career path, can you just briefly explain what honors is?
Yeah, so honors was part of my undergraduate degree. So when I'm saying undergraduate degree, I mean my Bachelor of Biomedical Science. An undergraduate will be the very first thing that you're eligible to do straight out of high school, sorry. So when I was doing my Bachelor of Biomedical Science, I had 3 years of coursework where I was doing lectures and tutorials and pracs, like I explained previously. So 3 years of that and exams at the end of every semester. And then the final year of my Bachelor of Biomedical Science was an honours year. So honours is basically a research focused year. So I was based in a lab that was interested in placenta, so in placental research, and I was looking at how the placenta and the uterus kind of talk to each other during pregnancy. So I did my research project, which is basically like having a full-time job. So I would go there every single day. I didn't have lectures outside of that anymore. It was just this one project. So I would go there every day and I would work with my supervisors to kind of do my experiments initially with quite a lot of supervision. You're not just kind of thrown into the lab and expected to know what to do. You have a lot of training because there's a lot of really technical experience that you need to kind of build up over time. So I had a lot of help initially from my supervisors with how to do everything in the lab. And then I would be a lot more kind of independent later on, and I would work on my own research project, and then I would write a thesis about that research project. So I kind of had an idea, and I had my methods that I did, and then I had my results that I would write about in my thesis.
Nice. It's kind of like a baby, right? It takes 9 months, and then you have your little thesis at the end of it.
Yeah, a very small
Um,
baby.
yeah, very well, a 50-page baby or so.
Yeah.
And then obviously a PhD is kind of that, but expanded. I kind of want to move on to your research now, and then if we have time, or if anyone has questions, we can kind of backtrack. But you've moved to Melbourne, which is awesome. After your PhD, why the move?
So I got to the end of my PhD, and generally there is kind of an expectation in scientific research in academia, I guess. When I say academia, I mean people who work in a university. So there's kind of an expectation in academia that you'll have a broad range of experience. So whether that be, working in Australia and working in Europe and working in America. I happened to finish my PhD in 2020. So there was no option to go overseas, which is fine with me because I'm quite happy to stay in Australia. And I knew that I had to kind of branch out of UQ a little bit because I I had been at UQ from all the way through my undergraduate, and then through my PhD as well. So I knew that I kind of needed to show that I could be in different environments and be productive in different places and not just kind of stick to my comfort zone, I guess, at UQ. And I met my my current boss, my supervisor now. I met her. She was my thesis examiner for my PhD, and she was working in a field that I was super interested in embryo development. And so I kind of approached her and asked if she had any jobs, and she did. She had money at the time, so it all kind of just fell into place, I guess, at the right time. So yeah, so I moved to Melbourne to specifically come to Monash as well, because Monash is, I guess, well known as a very good university, but also very good in the field that I'm interested in, which is in fertility and reproductive biology. So Monash has a very good reputation for that. So it seemed like a perfect fit.
Yeah, absolutely. And look, it doesn't hurt to ask people for opportunities, right? Look at where you are now.
Exactly. You can only get what you want from asking for it.
Mm-hmm. So now you're at Monash University and the Australian Regenerative Medicine Institute. I believe you have some slides to talk a little bit about your research. I'd love to hear it. So if you wouldn't mind, please, telling us what you do.
Yeah. Okay, so hopefully those are showing up for you now.
Yes, it is.
Okay, so my work is as a cell biologist. So a cell is made up of lots of different parts, such as they're held within a membrane, so that just holds the cell together. There's also a nucleus in the cell, so that's where all of the DNA is kept. They have different things inside the cell, but one of the things that kind of helps everything move around and stay where it is called cytoplasm. So that's kind of the fluid inside of the cell. So you can think of these as like a little fluid-filled sac. There are lots of different organelles, so we call them organelles. They're basically little mini organs inside the cell, and they have lots of different specialized functions. One that some of tonight's students may have heard of are mitochondria, so they're known as, the powerhouse of the cell. So they're involved in energy production. But cells actually have a sort of skeleton as well. So it's called the cell skeleton or the cytoskeleton, and that's kind of what gives the cell structure and gives it its shape. And one of the parts of the cytoskeleton that I'm particularly interested in are called microtubules. So microtubules are these long kind of filaments that are made up of lots of little parts that all polymerize together to form these long microtubule filaments. And they give the cell structure. Without microtubules, you see a cell just collapses, can't stay open. They help the cell divide when they're growing and dividing into more and more cells. They also help transport nutrients and different things around the cells. So if you need more mitochondria up one end, or if you need more protein of a certain type down the other end, the microtubules act kind of like a train network. So in this way, they give cells their function. And we work in the embryo, the mammalian embryo, especially using mouse embryos, because obviously human embryos are quite difficult to study. So we use mouse embryos as a very similar kind of model for embryo development. And we're interested in embryos because they go through these rounds of divisions and they get to a stage where they have these special cells that are called stem cells. So stem cells have these amazing properties to form any cell type in the body. So you can take these stem cells and you can form any sort of cell type that you see in an adult, whether that's eye cells or skin cells or bone cells. So we kind of want to know how this one cell can turn into all of the different cell types in an adult. And so, as I mentioned, we're really interested in the microtubules because they have all of these special functions, like acting like a transport network for moving things around the cell. helping in division, etc. So when we look at a cell in a mouse embryo, so this is roughly a 16 cell stage mouse embryo, and we can see here that it's got a nucleus which is purple in this image, and we've got microtubules as well. So we're really interested in understanding how these microtubules are arranged, and perhaps if we change their arrangement, would that change the type of cell that they become? Because microtubules look different in different cell types. So if we can change the microtubules, will they, instead of becoming a gut cell, for example, will they become a muscle cell or a nerve cell? We use, as I mentioned, mouse embryos to study this, but we also use human stem cells. And when we say stem cells, a lot of people think that we're deriving these from embryos. And actually, that's quite an old technology that's a little bit more, I guess, outdated in a way. The thing that we do now is we can actually take skin cells from adults, so donated skin cells. You can just take kind of a scraping from your skin anywhere, and we can undergo a reprogramming of these skin cells into a stem cell. So we call them induced pluripotent stem cells. So that's basically taking an adult cell, turning it back into its derivative of a stem cell. And we can use these stem cells to study all sorts of different things. We can keep them growing in dishes, and we can make them different colors, and we can get them to grow and get them to divide, and we can watch what happens. So, as I mentioned, we can grow these in the lab. So we can use microscopes, as I've got an image here. I spend a lot of time on the microscope, and I often have fluorescent labeling on my cells. So just to give you a kind of an idea what that looks like, we have live cells that we can label different parts of the cell, and then we can kind of see where they're going, see where they're moving, what shape they are, if they look different. And what we can do is maybe interfere with the growth of microtubules, so interfere with that cell skeleton. And we want to ask the question, if their cell skeleton, their microtubules, are different, would they not be stem cells anymore? Would they change into a different type of cell? So we can target microtubules. The kind of old school method of doing that is just by adding chemicals to the dish. However, that treats all of the cells in the dish. So we want to devise a different plan. We want to use light activation, where we take the cells and we specifically target microtubules when and where we choose. So we want to be really targeted with how we change the microtubules, so we're not just disrupting everything. And some of the ways that we can do that are using lasers. So you can see in this movie playing here, we see all of these kind of growing, they almost look like worms. We call them comets though, because they kind of look like shooting stars. So when you see them moving there, you can actually see microtubules growing in real time, and these are in live mouse cells. And then we can actually light activate a drug and make that growing microtubule stop. And we can do that in the embryo as well. So you can see the first panel in the middle there is all of the growing microtubules in an embryo. So that's kind of shaped like a sphere. And then if we take an image of a cross section through that sphere, we can see before and after we laser activate those drugs to interfere with microtubule growth. You can see that we can stop microtubule growth. So this gives us a really targeted approach to changing the microtubules. And so what we want to do with this research is kind of understand if we can change the structure of the cell or change the microtubule network, and see if we can actually change the fate of an embryo or the fate of a stem cell. So this might be really interesting for regenerative medicine, so using stem cells for regenerating different tissues in the body. Perhaps, we can influence a cell to become a muscle cell or become a nerve cell or become a skin cell. Say if you have a wound and you want to use stem cells to repair your wound, you might be able to use changing the cell structure to make more skin cells. We can also potentially influence embryo quality, something that I'm quite interested in is in making IVF more efficient and more successful, and perhaps in understanding why certain embryos don't develop. So if you're going through IVF and there's little success rate with the development of embryos, perhaps maybe their cell skeleton is defective, and we can see if there's anything we can do to kind of help give them a bit more help to develop. So that's my research, and so that's kind of what I want to do with my research there.
Such fascinating research and like beautiful, beautiful images. If you do have questions for Jess and her research, don't forget that you can pop those in the Q&A box down below, and those questions will be passed on. But I want to ask, what's a typical day for you as a research scientist? Are you just like constantly looking down the microscope or just shooting cells with lasers all day long? Like what do you do day to day?
You kind of hit the nail on the head there. So it's difficult to describe a typical day for me, because there kind of isn't really a typical day. I guess every kind of day is quite different, depending on where I'm at in my experiments. I spend a lot of time in the lab, so I spend a lot of time. We grow those stem cells ourselves, so I spend a lot of time feeding and growing stem cells, and helping them divide and helping them keep fresh and happy. That takes up a lot of my time, as does being on the microscope. So I spend a lot of time when we have those fluorescently labeled cells. It takes a lot of time to take really nice images of them. So I spend a lot of time on the microscope. I spend a lot of time also, so that's mostly kind of independent work where I'm by myself in the lab or by myself on the microscope. But actually now in this position, I spend a lot of time talking to other people. So that's something that's kind of new for me. As a postdoc, I'm now supervising my own students. So I have a student at the moment who's writing her thesis, and I'm helping her with how to write her thesis and kind of going through her draft and helping her improve her draft. So I spend a lot of time also reading and writing. And now that I'm working as a postdoctoral research fellow, I have a lot of kind of broader roles outside of my lab. So I'm part of a number of different committees in organising seminars. So this morning, for example, we had a seminar in our institute that I was hosting. So that's, again, talking to people and watching other people present their research and asking them questions on their research. So it's always very different, and there's always something going on. So there's a lot of time management that I need to do to kind of figure out how I'm going to fit in different experiments around different meetings that I have, and other commitments where other people need me. For example, my student. So I spend a lot of time also organizing my timetable, because I sort of need to keep everything managed quite closely. But yeah, it's great because every day is quite different, so it's hard to get bored.
Yeah. And I guess also changing it up, like, you mentioned seminars and meetings and things like that. And I think a really important part of being a researcher, whether you're a student or further along in your career like you are now, you need to share your work with others and talk to other people. So researchers often travel to conferences and things like that. What were the opportunities for you, maybe as a student and now? And I know times have changed with a pandemic, but what are the travel opportunities that have come your way because of science?
Yeah, travel is one of the huge perks of this career. It's very easy to get funding to go to different conferences around the world. So when I was a PhD student, I got funding from the Faculty of Medicine, where I was based at UQ. They had money set aside for PhD students who wanted to travel internationally. So I signed up for a conference in France, and I went to a little French town called La Rochelle, which was absolutely beautiful. So I flew into Paris, and then I caught the train out to La Rochelle to this beautiful kind of seaside town in France. And I spent about a week there going to this conference where there was scientists from all over the world, particularly one who I was extremely starstruck to meet. She was one of the kind of biggest influences on my PhD project, because a lot of her papers were, very interesting and very closely related to my work. So I got to meet her, which was super exciting. And I actually presented a poster over there. So when you go to a conference, you can have an oral presentation, which will be like a verbal presentation where you show slides and you talk about your work, or you can make a poster on your research. So I had a poster presentation at the conference, and I got to travel to France, and it was amazing. Since then, I've traveled to Washington, DC in December last year. Obviously, COVID kind of slowed travel down for everyone. So I'm hoping that we can get back up and running with it again, because, going to Washington, DC in December last year was also very, very, very cool. A lot of domestic travel as well. So I've been all over Australia, the Hunter Valley, been to Adelaide, I've been to Perth, I've been, all over Australia as well, talking about my research, which is very cool. It's one of the major perks of being a scientist, I think.
Yeah, absolutely a big perk. But is that the thing that you like most about being a researcher? Like, is there one thing that you're just like, yes, this is what I do it for?
I think it's more to do with the problem solving. I really like problem solving and kind of needing to be creative, I think, with, we might have a question and we say, well, how do we actually study that? Our question is, how do the microtubules shape cell identity? Well, how do we even begin to look into that? So that I find really interesting. And it's always, it's always a challenge, which I think is a good thing, because otherwise it would be a little bit boring, because, I want something that's challenging and that you constantly have to be thinking about, how can I do this? How can I solve this problem? So I think that's kind of my favorite thing about being a scientist, is using my creativity to try to solve problems.
And you're constantly asking questions, and the science sort of never stops and never ends. So it might be difficult to move on from the project that you're working on. But what do you want to do next? Like, are you happy where you are? Do you want to move on to some other city?
Yeah, that's I don't know. I am happy where I am. But I'm always kind of looking for, what's the next step? Where do I want to go next? I'm sort of thinking the next major step for my career would be to get some of my work that I'm doing now published. That's something that's super, super important for scientists, is publishing their research. And that can take quite a while. It can be quite a long process to get it to publication. So that's kind of my focus for the moment. The next kind of major milestone I want to hit is getting independent funding for my research. So just briefly, so the funding has gone from the government to my supervisor, because she applied for grants, and that was what has been able to fund my position, so pay my salary and pay for my research. So I'm employed by Monash University, but kind of under my supervisor, who was awarded funding money from the government in a grant. So the next kind of major step for me is in applying for my own research funding, so that I can kind of pursue my ideas independently. So that would be moving into a group leader position myself, so getting my own students and potentially having my own kind of staff members that work for me. But that can take a while. So I don't know if that's really like a 5-year goal. Maybe that's a longer term goal than 5 years, but it's all about kind of working towards, the long-term goal in shorter, more achievable chunks.
I feel like I've taken on the role of one of your mentors, because you mentioned at the start, they ask you, where do you see yourself in 5 years?
Yeah.
So I guess you've kind of answered that. But what impact do you want to make with your research and your career?
Yeah, I think there are a number of different ways that you can make, impact as a scientist. Obviously, there's the really big discoveries that are Nobel Prize worthy, but those aren't necessarily a common outcome for most scientists. But I think it's just as important to make kind of incremental changes to our understanding of the world. So, I've always been super fascinated by what makes us human. How do we go from being an embryo to being a person? What makes a cell act in the way that it does? I've always been super fascinated by these really fundamental kinds of questions. I find them almost like a philosophical question in a way, like what makes us human. So I think broadening our understanding of those really fundamental things of what makes us human and what makes us alive, I think that would be something that I want to leave behind when I, am at the end of my career. That would be kind of the impact that I would want to make is kind of broadening our understanding of how we are the way that we are. I think you can also make an impact in different ways. So, you can make an impact on students. You can, mentor students who are in your lab, or you can mentor, students who are in a different lab in the same institute as you. So I want to make an impact on students and potentially, hopefully, try and make scientific research kind of a more inclusive place and a more, I guess, friendly place. It doesn't need to be so serious all the time. So I hope that, my impact can be in kind of making it a happier place for students and a happier kind of more inclusive place for everyone in the institute.
Yeah, well, I mean, hopefully you're already making a big impact for the students who are listening in tonight. I have one final question that I want to ask you. I have a long list, but one question that I want to ask you before I start taking some audience questions. So remember, if you do have a question for Jess, type it out, pop it in the chat, and I'll ask it. My question is, you've made lots of risks, made lots of big or taken lots of risks, I should say, and made lots of big decisions like moving from rural Queensland to Brisbane to Melbourne. Do you even do a PhD? All those sorts of big decisions that you've had to make. Does it get any easier as you progress through your career to think about, what's next?
Yeah, definitely. Definitely. I think you become kind of more comfortable with the unknown. Although saying that, I kind of look back at when I was a high school student and I decided to move to Brisbane and study science. I sort of think, God, that was brave. Like, what was I thinking? But I think you definitely get more comfortable taking those kind of risks, I guess. You don't necessarily have to have it all figured out. That's something that I've learned along the way, is that a lot of the time I sort of made decisions not really knowing what was going to come of that, choosing which degree I was going to do or choosing whether or not to do a PhD. I didn't really know how it was going to end up. But I think if I was indecisive and if I was a bit too afraid to make the call, I think then I would have missed out on a lot of, really amazing experiences. So I think especially when you're younger, you can sort of feel like you need to have it all figured out by a certain age, or you look at adults and you think, wow, they've really got it all figured out. They know what they're doing. A little
Do
secret
they?
is
Do they?
we don't. We don't at all. Yeah, so I mean, no one has it figured out. I can talk to mentors now that are full professors, and they're, you look at them and you think, wow, they're amazing, but they don't have it figured out either. They're just as scared as the rest of us. So I think, I think being scared is normal, and I think, being unsure is completely normal as well. And if you're not 100% sure on your decision, just do it anyway. It can be really fun. I think don't let indecision stop you from kind of pursuing something that you want to pursue, because you don't have to have it all figured out. You can change your mind. You can change direction. I've done it multiple times. It's fine. You figure it
Yeah.
out.
As have I. And look, we're both fine, right?
Totally fine.
So yeah, you can definitely, try lots of things and then, you work out what you like and what you don't like, and you can change your mind as you go. I'm
Exactly.
going to move on to some questions, and the first questions that are coming through are more specifically about your research, Jess. So, Alex asks, if you're taking cells from the human body, does it matter, the area or the tissue that you're taking it from? Does that impact the quality of the stem cells that you can then make?
Yeah, it kind of does. Not necessarily different areas of the body, but you have kind of different, I guess, more of like a hierarchy of stem cells. So when I spoke about the embryos, so when that embryo is a one cell, so when it's completely just one cell, just an egg and a sperm mixed together, that's what's called totipotent, a totipotent stem cell. So totipotent means totally potent. So it has lots of potency, meaning that it can become absolutely anything. So anything in the body, anything in the placenta, anything in the amniotic sac, everything. Whereas when after it goes through a couple of rounds of division, they're then called pluripotent, so plural potency. So that can become everything in the embryo. When you have stem cells in an adult's body, those are called multipotent stem cells. So those are stem cells, for example, you have stem cells in your bone marrow that are multipotent stem cells that can form all of the different blood cell types, but they can't become skin cells or they can't become brain cells. They can only become blood cells. So I guess when you ask, does that affect the quality of the stem cells? I guess it kind of does, because you can't take bone marrow stem cells and make a new eyeball, for example. So they're kind of limited when you're getting the stem cells from different areas. They're only able to become their kind of local tissue that they're from.
Yeah. And on that, Lavanya's asking whether it's possible to take totipotent stem cells from another, but maybe a similar species to humans. But can you take them from another species and work out what's going on and turn it
Yeah,
into
I
maybe?
mean, that's a great question, and it would be really cool if we could. Um, Not that I know of, though. Not that I know of. I think, you would have the same kind of problems of, rejection from your body. Your body would recognize that as kind of a foreign thing, so you wouldn't be able to necessarily take, maybe from like a monkey or a mouse or something kind of similar. You wouldn't necessarily be able to just put them into your body and have your body kind of accept them as human. I think your body would probably reject those.
Yeah, absolutely. You're kind of touching on my field now because I study the immune system and immunology. But the human body can even reject other human body parts, like hearts and things that are received from other humans, let alone another animal. So, yeah,
Yeah.
I guess it might be a bit tricky.
Yeah, so that's what's really cool about the induced pluripotent stem cells that I spoke about. So taking skin cells and then turning them back into pluripotent stem cells that can become any cell in the body. So then you can actually take skin cells from yourself, turn them back into pluripotent stem cells, and then use them to form any tissue that you need. So that would kind of be a way around, instead of using cells from a different species, you can use your own cells and actually make them form any kind of different cell that you need.
So that's really part of personalized medicine, right? Would the way that you do that and turn a skin cell from a person into a pluripotent stem cell, would that be the same no matter who it is? And so you can at least use the same technique or same technologies across different people?
Yeah, it would. So they would undergo the same kind of reprogramming process. So they would give the same factors to the skin cell to turn it into the stem cell. In terms of how they actually use those stem cells then in the body, that's kind of an active area of research now that people are trying to figure out. We have this idea that we can use skin cells, turn them into stem cells, and then use those stem cells to, repopulate, say, a wound, for example, or maybe, you want to grow a new body part. How do we actually do that? So that's quite an interesting area in kind of the biotechnology sphere, where they're looking at different, biomaterials that you can have mesh networks that you put the cells into, and then you put that mesh into the human body, and it helps kind of hold everything in place. And so that's kind of a very cool. I don't know as much about biotechnology, but I find that area is very interesting in looking at all of the different materials you can use to actually use those stem cells in a real patient.
That's really, really exciting. Like, especially knowing that, it can be so hard to get organs when people need them. And, you mentioned wound healing as well. And Nellie asks if you've ever used fish in your studies to analyze their cells. And I know that there are certainly some aquatic species that have great regenerative capacities. Like, do you look at fish at all?
I personally don't, but because I'm based in the Australian Regenerative Medicine Institute, there are a number of people, and it's great that you asked that question because they are such a regenerative species, or I guess family of animals. So a lot of fish and aquatic animals can actually completely regrow limbs, or they can, they have a much, much better ability to regenerate after injury than humans do. So they're actually used to kind of understand, well, why are fish able to regenerate a limb and we're not able to? So I personally don't use fish, but where we are at the Australian Regenerative Medicine Institute, there's a huge aquatic centre where they have lots and lots of different types of fish and sharks as well. So they use those fish and sharks to kind of study, how their regeneration works and if we could potentially have the same regenerative capacity as humans. So there's some very, very cool fluorescent fish that they use for those kinds of studies, and they're really, really interesting. But personally, I haven't, unfortunately.
I've seen the facility there at ARMI, or the Australian Regenerative Medicine Institute. It's definitely worth checking out if you get a chance to as a student. Caitlin asks, what are the best alternative species to study cells instead of humans? Because obviously we can't do everything in humans when it comes to research. So what are some good animal models or animals that you can use?
Yeah, so I guess it depends what your application is. For me personally, I use mouse embryos and mouse cells. That's because I'm interested in, the fundamental cell biology. So our cells are quite similar to mouse cells because they're a species of mammal, and they're a lot easier to get than human cells. They're a lot easier to grow. Obviously, we can't, work on human embryos, and we can work on mouse embryos. So those are a lot more readily available to us. It depends, I guess, what your application is. One of the other students asked about fish. So fish, like I mentioned, have, a great regenerative capacity. So if you're kind of looking at, for example, there's a lot of research in our institute on muscle repair. So if muscle is injured, how does that actually repair? Fish are really great for that because fish, particularly small ones, are quite transparent. So you can actually use a microscope to look inside the fish and you can see everything kind of happening, and you can see the muscle cells that you're interested in, and you can still have the fish alive and be, swimming around. So it totally depends what your application is, really.
Jess, you've been doing such a great job communicating, really complex topics. And I think that's something you've been doing throughout your career, like, conveying complex science information to a range of audiences. You're chatting to the students now, you've been a university tutor, you've had to talk to clinicians as well. So communication is one, but what are some of the more, I guess, translational, or we call them soft skills, that you pick up as a science student?
Yeah, so I guess communication is definitely one of them. I think also you need to kind of have a certain level of empathy, I think. Um, Whether that be empathy for, if a student is struggling, you can kind of know how to help them, but also empathy in understanding other people's perspectives. So when you're, I guess, communicating to someone, you need to kind of understand what they already know. So if I'm talking to a high school student versus if I'm talking at a conference, or if I'm talking to, a clinician like an embryologist, I'll talk to them in a different way because they have a different background and they have a different understanding of what I do. So I think having that kind of ability to see things from other people's perspectives, or I guess try and guess what their perspective is and try and understand what they already know and how you can explain something in a way that they understand. I think also kind of another soft skill would be, obviously I've mentioned problem solving, but also being resourceful, because oftentimes, you might have all of these incredible ideas and you just don't have funding to do them, or you don't maybe have, say you want to do a particular experiment that needs a type of microscope that you don't have, or a type of chemical that you don't have. So you need to kind of think about, okay, well, how can I do this a different way? So it's kind of in understanding your resources, um, and also kind of how to reach out for help, I think, is a big one. I'm always emailing people, asking for help, asking them to explain something to me. I think that that's a really important skill, is kind of asking for help and asking people for their advice. And that's something that I do all the time. Whenever I have a problem or I'm not sure about something, I think, okay, well, who do I know that would know the answer to this, and how can I'll write them an email and I'll how can I explain what I'm trying to say. And yeah, I think that's a big skill that I'll take with me wherever I end up, is asking people for help.
I think we've always got to remember, like, it's okay to ask for
Yeah,
help when you need it.
definitely.
Speaking of resourcefulness and resources at your disposal, I know that at a point in your career, you moved to an entirely new lab and you had to set everything up. What is it like setting up a lab from scratch?
Yeah, it's very complicated. I worked so when I finished my undergraduate, my Bachelor of Biomedical Science, I kind of got to the end of my honours year and I wasn't really sure if I wanted to stay in research. I think I was just a bit stressed at the end of my honours year, and I kind of I wasn't really sure what I wanted to do, and I felt like I needed more time to decide. So I actually worked as a tutor for a while at UQ, but then I took on a research assistant role. So a research assistant is basically someone who, you could call them like a lab manager essentially. They're kind of a technician, and they do all of the ordering and they do all of the preparation for different experiments. Say if someone needs a lot of their samples tested, they say, here are the samples, this is how I want you to do it, and you go and do the testing and then you bring them back the results. So I worked as a research assistant for 3 years. The second lab that I worked in, I had to set everything up from scratch, because my boss had moved over from London. So he moved from London, and started working at UQ. So pretty much he had funding money, but he had literally nothing in the lab. It was completely an empty room, and so I had to kind of learn on the go of, what do we actually need in a lab? Where do you begin? So it was a lot of, again, asking for help, asking other people, where did you get those tubes from? Where did you get this thing from? Where did you get this chemical from? We need that same thing. How did you find that? So it was a lot of time talking to suppliers and buying things like microscopes that I had never bought before, and it was $500,000 worth of microscopes. So I didn't really know if I was buying the right thing, but I think I got it all right, and it all worked fine. So yeah, it was a lot of kind of just learning things as I go, and trying to find the right reagents and equipment for the lab. So it was
Yeah.
interesting. It's quite a challenge.
And then thanks to you, well, you asked for help, which was great. And then once you succeeded, your lab had all the resources that you needed. So excellent.
Yeah.
One of the students asks, what would you say is the best part of studying in science? I know that you talked about the things you liked as being a researcher, but if you sort of rewind a little bit to, as you study science, what's the best thing?
I think there's so many different aspects to science. Like I was saying, I thought I was maybe interested in neuroscience. Turns out that I couldn't look at maps of brains and understand all of the different areas. That completely confused me. I just couldn't wrap my head around it. So I think, yeah, I think the best part of studying in science is probably that there's always something different. There's always something to learn, and there's so many creative people who are writing papers and showing their work, and there's always someone doing something really cool and interesting. So I think, yeah, there's lots of different aspects to science, and if you're not enjoying, say you don't really like chemistry, for example, you don't have to work in a chemistry related field. Or say you don't like genetics, you don't have to do genetics. So you can kind of make it what you enjoy and what you want to do more of. I really love cell biology, and I really love working with microscopes. So that's kind of where I spent most of my time and spent most of my focus, because that's what I enjoy. So I think that's kind of the best thing in studying science is if you're not enjoying one part of it, there's so many more different fields of science that you can be interested in, and you'll find one that you like best.
Yeah, absolutely. And speaking of making it yours and pursuing what it is that you want to do, I just want to know your experience in bringing other sort of interests into science, whether you've done it yourself. Lavanya's asking specifically about literature, and you might not necessarily know about the combo of literature and science, but what's your experience with bringing different interests together?
I guess it depends. I guess what kind of literature you're talking about. Um, you know, a lot of science is reading and writing, and if you're interested in reading and writing, there's plenty of that in a science career, in a research career. You can also work as like a scientific writer, for example, so you can assist other people in writing their papers. So to answer your question about bringing other fields in, I guess I've probably taken a more traditional approach to my career, in that I'm very much kind of a fundamental cell biologist. But there's heaps of other careers. I mean, I'm sure you can speak about your own experience in working in communications. So I think you can definitely, especially if you're interested in education, you can kind of incorporate an education or communications role in a science career. You can do other things as well, like you can work in, making policies and advising governments. Say if you're interested in like environmental policy or policies in terms of different medical, biomedical aspects of policy. I think whatever you're interested in, there's a way that you can make it happen.
Absolutely. And as you say, you're a traditional, maybe a more traditional research scientist, but there are so many different things that you can do, as you've pointed out, Jess. And no, I don't want to totally hijack this, but I might just add that I really loved music in high school, and I now pair music and science. So it's definitely possible. Like, I get paid to write songs and sing songs about science. So, you never know where your interests will lead, and you can certainly mix them together.
Exactly.
Amber is asking how you manage the workload in your undergraduate degree, so the first 4 years of uni. Did you find the content a lot harder than high school, perhaps?
I did. It is harder than high school, I won't lie to you. But I think, because it was something that I was so interested in, that kind of made it a bit easier to spend a lot of time studying. So I was pretty diligent in going to all of my lectures, but I still found, whenever it came to exam time, I was still going over the lecture material that I had been taught weeks ago and was like, God, I don't remember this at all. So it is quite a lot harder than high school in terms of the content. I think, everyone struggles with the workload at uni from time to time. I think if you're struggling quite a lot, there are often there are different options available. You can drop certain subjects, or you can take it part time if that's something that you need to work on. I think at uni there's also a lot more opportunity to work with your peers. So, if you have an assignment, you can meet someone at the library and you can kind of work on it together. Obviously, submitting things separately and doing your own work, but I think there's definitely opportunity to kind of get the help that you need. How I manage the workload, I was quite lucky in that I managed to get a scholarship for my first year of uni, so I didn't kind of have to worry about working on the side. But it's definitely doable. I had a number of different friends who had jobs, like casual jobs, working in a store or working in a restaurant. I myself worked in a gift shop when I was in second and third year. I also worked in a cafe as well. So you can also work at the same time. I think it's kind of just about managing your time really well, and trying to make the most of your time while you're at uni. So if you're at uni, you want to make the most of your time and get the work done that you can, because that can kind of help you in getting ahead if you're struggling a bit.
Yeah. And one of the questions, I think you've kind of answered already what subjects you did in high school, because someone's asking specifically about maths methods, but that's not really a thing that you did in Queensland. So perhaps we might leave that one. But a completely different question. You've shared what you're researching now, and this might be a difficult question because what you research is close to your heart. In the end, if you weren't researching embryonic development, what would you be interested in? What would you be studying?
If I wasn't a scientist at all,
I think it's more about the research. So, like, if you pick something else to research.
Yeah, good question. I haven't actually really thought about that. I think, I'm interested in kind of cell biology in general. I think, if I wasn't in embryo development, I would like to be in maybe cancer cell biology, or, just how cells actually work, how they look on the inside. I find that really fascinating. Something that I mentioned before that I, kind of have a bit of an interest in as well is, the biotechnology aspect of kind of using stem cells. There are a number of different startup companies who are even based in Melbourne, but based all over the world, that they're trying to use stem cells for regeneration. So if you need them to regenerate a wound or regenerate damage in the body, I think that that's quite an interesting area. So using different plastics or using different biomaterials or how do you actually use those stem cells in the body. I think that that's quite an interesting area. I think cancer cell biology is also quite cool because, you know, I'm currently working on how cells work, but a lot of cancer cell biology is when cells are working incorrectly. So how does that actually happen? So I think maybe that would be kind of an alternative to what I'm working in now.
Hmm. This is an interesting question. It's more about like studying techniques, I guess. Biology is a completely different language. There are so many words in biology, and you've introduced us to a lot of new terms tonight. What are some of the ways that you've used that kind of help you remember all of these new terms and the function of everything?
Yeah, I think it's interesting, and that's something that I kind of learned at uni, I guess, was what kind of learner I am. So I found that I was a very visual learner. If I sat in a lecture and listened to the lecturer speak to the class, I could walk out of that room and not remember a single thing. Um, Whereas if I took notes as they were speaking, or if I drew, especially if I would draw something, I found that that helped me remember things. So I think that that's kind of something that I learned, later in high school and while I was at uni was I'm quite a visual learner. Some people aren't visual learners. Maybe you work better if you listen to the recording of a lecture in an earpiece and you just have it playing in the background and you can kind of take the information in that way. That doesn't work for me. So some of the techniques that I use, and I still use them to this day. Like I still hand write out notes in seminars, or I still, when I'm trying to think of ideas, I'll get a pen and paper and I'll just kind of draw. Say if I'm looking at something inside a cell and I'm trying to think, oh, how is this working? I'll try and draw, this piece moves over here, and I'll have lots of arrows. And so I think you've got to figure out what works for you. But remembering the words and their functions, for me, it was just making diagrams, writing the words, putting them in the right place on the diagram, and kind of having that visual memory of where it goes, or, the arrow of it goes over here or it does this thing. That kind of helped me in remembering the words. You can also, I often use this word sounds like this thing. So, like, if it sounds like a name of someone I know, that's really helpful. Or if it sounds like a place I've been, I find that having that association in my mind kind of helps me remember the word in different ways. Or if I make like a funny kind of sounding thing out of the word, just whatever way you can try to remember it. It's
Yeah,
often quite
thanks,
weird.
Jess. Learning how you learn best, it's a very important thing. We're running out of time, but I just have one final question for you. You've got all these young, impressionable students who are wanting to pursue science. In under 60 seconds, I've got a timer up. In under 60 seconds, what is your one piece of advice that you'd give to students who are thinking about studying science?
I think if you're interested in science, absolutely go for it. I think it can be really challenging at times. It's a lot of work and it's a lot of studying, but I think if you're interested in it, it's such a rewarding career. I often feel like I don't have a real job. I kind of get to play around in the lab all the time and play around on the microscope, and I'm playing with stem cells and I'm playing with embryos. I feel like I don't really have a real job. So I think, yeah, my advice would be if you're interested in it, it can be really, really cool and it can be really, really fun. And you can kind of make it what you want. Like I was saying earlier, there's so many different aspects to science. You can find something that's your kind of niche favorite thing that you can't stop thinking about. And you can just make that your own little world and live in that bubble.
Amazing. Oh, 58 seconds. Well done.
Perfect.
Well, just on behalf of everyone here, Jess, I just want to thank you. So I know we can't see everyone on the call, but I'm just going to clap Dr. Jessica Greaney, and hopefully you can all, join my sentiment in thanking Jess. Um, I just want to remind you all that this Superbio series was developed by Monash Tech School with this very, very grateful support of the Victorian Department of Education, and it's delivered by the Convergence Science Network. So again, claps, claps, claps. So thank you so much to all three of those partners for making this series possible. The next Superbio Masterclass will be held on Wednesday, the 2nd of August at 6:30 p.m. again, and will feature Dr. Kirsten Carey, a research fellow at the RNA Biology and Cancer Lab at the Peter MacCallum Cancer Centre, who's developing new cancer therapeutics. So very different, but it's amazing to be hearing from Jess, and if you tune into Kirsten as well, like hearing from such amazing biologists. So I encourage you all to save the date, Wednesday, the 2nd of August, and look out for the event details in the coming months. Also, as Stephanie was asking a question in the chat about, what can you do to explore many different courses and opportunities? Come to things like this. Another thing that you can do is the Convergence Science Network has an extensive video library, and I definitely encourage you to visit it and discover some more amazing science talks. I believe we do have the links to show you somewhere on a screen. I've definitely learned so many fascinating things from previous talks at the Convergence Science Network, and that includes fun facts to bring up in conversation. So I definitely recommend checking them out. So we can't see the links now. I guess maybe when I stop talking, you'll be able to see them. So thank you very much, everyone, for coming along. Thank you to all the people behind the scenes, like I said, from the Convergence Science Network, from the Department of Education, as well as Monash Tech School. And hopefully we'll see you next time with all of these amazing partners behind us. It's going to be a good one. So see you there.